Hello students, I am teacher Svetlana and in our today's session we will see about genomics and the Human Genome Project, which is covered in the chapter Molecular Basis of Inheritance. So let's get started. Students, the term genome was first introduced by H. Wingler in 1920. He says that it is the total genetic constituent of an organism. Alternatively, it is a complete copy of the genetic information, that is the DNA, or one complete set of the chromosome, either the monoploid or the haploid state of an organism. And the term genomics, this was coined by T.H. Roderick in 1986. Genomics is the study of genomes through analysis, sequencing and mapping of the genes along with the study of its function. So in genomics, basically the entire genome is analyzed. It is sequenced and mapping of the gene, that is the exact location of the genes, is identified and its functions are studied. Genomic studies is classified in two types. Structural genomics, it involves mapping, sequencing and analysis of the genome, and functional genomics. It deals with the study of the functions of all the gene sequences and their expressions in organisms. So here you study the genome and you analyze their locations, and in functional genomics you try to understand the function of each and every gene, how it is expressed in an organism and how the expression can be regulated. Here you can have the difference between the structural genomics and functional genomics at a glance. Structural genomics, it aims to determine the structure of every protein encoded by the genome, identifies novel protein folds and 3D structures for better understanding of the functions of these proteins. Whereas in functional genomics, it aims to collect and use the data from sequencing for describing the genes and the protein functions. Functions of the genes and non-gene sequences in the genome are understood, and then the gene and protein interactions are observed and studied. You also have a look at the genotypes, phenotypes of an organism by understanding the functional genomics concepts. Application of genomics. Structural and functional genomics is used for different purposes in the involvement of crop plants, human health and livestock. The knowledge and understanding acquired from genomics research can be applied in a number of different sectors. Now, these sectors include medicine, biotechnology, and social sciences. It helps in the treatment of genetic disorders through gene therapy. So, the list is countless. I have listed a few of these applications for you. Genomics is used in agriculture to develop transgenic crops having more desirable characters. Genetic markers developed in genomics have applications in forensic analysis. Genomics can lead to introduce new genes in microbes to produce enzymes, therapeutic proteins, and even biofuels. Have a look at this chart here. Genomics and its applications in livestock biodiversity, in plant genetic resources, animal health and vaccine development, mutation breedings for crop improvement. So even if you take one such factor and try to go in a little bit more detail, you will see applications of genomics in crop improvement. You will see why it is used and what information does it provide. It provides information about the genome size, gene number, gene mapping, gene sequencing, evolution of crop plants, gene cloning, identification of DNA markers, markers assisted selection, transgenic breeding, constructions of linkage maps, QTL mapping. So the list goes endless even in one particular field. So you have such number of fields where genomics has its use. Here also I have put the broader applications of genomics. They can be used for screening the newborn, then for prenatal testings, then they are used for monitoring disease and their occurrences, then prognosis and therapeutic decisions, disease susceptibilities, screening and diagnosis. So this is the entire medical application of genomics. Now students, let us have a look at the Human Genome Project. The Human Genome Project was initiated in 1990 under the International Administration of the Human Genome Organization, HUGO. This project was coordinated by the US Department of Energy and National Institute of Health. Additional contributors included universities across the United States and international partners in the United Kingdom, France, Germany, Japan, India, and China. The Human Genome Project formally began in 1990 and was completed in 2003. The Human Genome Project is a multinational research project to determine the genome structure of humans. Now students, let us have a look at the main aim of the Human Genome Project, the reason why multinational research project was carried on. Now among the main aims we have identify all the genes in human DNA. So this was one important aim, to identify the genes in the human DNA, map the positions of the gene on all the human chromosomes. So mapping is finding the exact location of the gene on the chromosome. Determine the sequences of the chemical base pairs that make up the human DNA. So at the nucleotide level, they wanted to know each and every nucleotide that comes together to form the human DNA. Develop suitable technological tools for analysis and subsequent use of the genome. Store the DNA information in suitable database for easy retrieval and analysis. This data they wanted to store in a suitable place. So anyone who wanted to access the data could use it for the further studies. Make the technological and genome information available to all interested parties, including biotechnologists, industrialists, pharmacists, and the medical professions. Address the ethical, legal, and social issues that may arise from the project. So students, to put it in a nutshell, overall the Human Genome Project had these aims, that is mapping the entire human genome at the level of the nucleotide sequences, store the same information and in the form of a database, and develop tools and techniques for analysis of this data, and they should be able to transfer these related technologies and informations to the private sectors such as industrials and all its stakeholders like the biotechnologists, pharmacists, the medical field. So this was the main aim of the project, and also take care of the legal, ethical, and social issues that may arise because of this project. Now students, let us have a look at the findings of the Human Genome Project. The Human Genome Project was closely associated with rapid development of a new area of biology called as bioinformatics. The work of Human Genome Project has allowed researchers to begin to understand the blueprint in building and construction of the human genome. So the blueprint is the actual basis on which the entire DNA is constructed. As researchers learn more about the functions of the genes and proteins, this knowledge will have a major impact in the field like medicine, biotechnology, and the life sciences. Therefore, the Human Genome Project is very important. Now, why this blueprint is required? Like when a particular say building or a site is constructed, first a map is drawn or a picture is drawn to give you an idea about precisely where what will be located, which we normally call as the architect plan. So that architect plan will decide where what has to be constructed. So similarly, the blueprint is the nucleotide level information about our DNA, how our genome is constructed. So by understanding that, we will be able to understand the function of the genes, the proteins that they code for, and this will help us to give a major impact in the fields of medicine. For example, we talk about diabetes, we require the protein insulin, which plays a very important role there. So what does that insulin do? It allows the entry of glucose into the cells. So person suffering from diabetes, this particular protein is not produced. So if there could be any modification done at gene level, then a person may not suffer from such disorder. So this is how Human Genome Project is going to help identify genetic disorders right at the gene level. Now, this Human Genome Project was to provide the complete and accurate sequence of 3 billion DNA base pairs that make up the human genome, and to find out the estimated number of human genes. Now, about 33,000 genes have been estimated to be present in humans. So this is a remarkable achievement of the Human Genome Project. Students, this project also aimed to sequence the genome of several other organisms such as bacteria like E. coli, then we have Saccharomyces cerevisiae, that is yeast, then Drosophila, that is fruit fly, and in plants we had rice, and of course about mouse. So complete genome sequence of these model organisms will be useful for comparative studies that will allow researchers to study gene functions in these organisms. Now, in addition to this, I would like to say that the secret of our complexity may lie not in the number of our genes, but how we use them. Now, it will lead us to the understanding of gene structure and functions in other species. Since we possess many of these genes same as those in flies, roundworm and mice, such studies will lead to a greater understanding of human evolution also. Students, have a look at this image. This image is called as the Human Genome Timeline from 1990 to 2003. If you have a look here, at 1990, the Human Genome Project was first launched in USA. Then you have in 1991, the first US Genome Center was established. Then in 1992, we had second generation human genetic maps that were developed. Then we move further into the timeline where you will find new 5 years plan. In 1993, a new 5 years plan for the Human Genome Project in the US was published. Then in 1994, the Human Genome Project human genetic mapping goal was achieved. Then if you have a closer look, you will see in the timeline different milestones were achieved, which went on adding to the success story of the Human Genome Project. Like in 1998, incorporation of 30,000 genes in the human genome map was done. Then you have in 1999, the full-scale human sequencing began, and it moved and the story continued. In 2001, the draft version of human genome sequence was published, and then in 2002, draft version of mouse genome sequence completed and was published, and finally in 2003, the finished version of the human genome sequence was complete. SGP ends with all goals achieved. So all the goals were achieved, and yet the further research is to be continued. But so far, the entire timeline is an interesting way to understand the Human Genome Project. Thank you, students, for participating in this lecture. We will meet soon again with the next topic. Till then, take care, be safe, and keep learning.